everyone, and welcome you to the launch of our exciting new webinar cycle, this time entitled Transition from Pesticides to Biopesticides. Webinars that are brought to you by GROW, which is a project that is part of the EIT Food, which Food4Sustainability is a proud partner. I am Claudia Costa. I'm the Academy Director at Food4Sustainability, and I will be moderating this series as we delve into one of the most pressing topics in modern agriculture, the question of how we can shift from conventional pesticides towards more sustainable, eco-friendly biopesticide solution. And we have prepared this cycle to explore the science, innovation, and regulatory frameworks driving this transition. And over the coming sessions, we'll hear from leading experts who will be sharing their insights on how biopesticides can help us tackle these global challenges such as soil health degradation, biodiversity loss, and the need for safer food production systems. And to kick off this discussion, we have the pleasure to have with us three distinguished speakers. And I will start by introducing Joana Cruz. She's the Director of Soil Health and Function Laboratory at Food4Sustainability. And Joana will provide an overview on how biopesticides interact with soil microbes and their role in promoting healthier ecosystems. Joana, thank you so much for joining. We had to change our agenda at a very short minute, and we are truly grateful for your availability. Also, we have the pleasure to have with us Mojgan Rabiey. Mojgan, she's an assistant professor at the University of Warwick. As we know, it's in the UK. And Mojgan will share a cutting-edge research on microbial biopesticides and their potential to replace chemical pesticides while maintaining crop protection efficacy. And we will also have the pleasure to have with us Marisé Borja. She's the manager for SynTech Global Regulatory Affairs Business Development. And Marisé will discuss the regulatory landscape for biopesticides and how business can navigate this transition effectively. And thank you so much for being here. And as we all know, these webinar cycles are not just about learning, it's about sparkling conversations that actually lead to action. Whether you're a researcher, you're a farmer, a policymaker, or if you're simply passionate about sustainable agriculture, we hope that these sessions inspire new ideas and for sure, new collaborations. And I will also take a minute just to remind you that registrations are now live for our upcoming Soil Health Summer School. If you don't know, this is a comprehensive program that invites you to embark on an in-depth exploration of soil microbiomes, delving into their transformative impact on planet health. Also, sustainable diets, drawing attention that if we want to pursue a diet based on sustainable principles, we need to look backwards to how our food is produced. So the pioneering strategies for sustainable agriculture. And as you know, this school is going to be mentored by renowned experts. So you will gain not only valuable insights, but also the chance to develop your own innovative projects. So again, if you're a student, a farmer, an agricultural professional, or simply passionate about soil health, I think this is the summer school for us. So register and join us later this year, and I can promise that you'll learn a lot and you'll also have fun. So thank you all for joining us today. Let's then start our session, and we start as we always start. So with a warm-up on Slido, which the team has already prepared for you. So if you don't know by now, Slido. You can either browse Slido on your computer. Here we have. So you can type www.slido.com or just scan these QR codes. Let's wait. Let's allow everyone in Slido and let's see what questions the team. One of these days, we're going to start to have some prizes on Slido. Right, team? For the best participants, the one that answers faster, because it's always very rewarding to see you all taking part in this in our Slido activities. So Soraya, I think you're in charge there. You're at the driving wheel. No, Soraya just nodded. I think it's Rita. Rita, thank you. So we have the first question. So biocontrol interventions reduce crop damage by what percentage? So if we start to look at biocontrol interventions, we are likely to reduce the impact of pests and diseases. What is the percentage that we are looking so far? And I have to say that, of course, all the questions that we bring you here, they are based on scientific evidence. So we don't come up with these questions. And this one was made on are the figures from a recent meta-analysis study that showed that biocontrol interventions reduce crop damage by we have a tie. Can I have a vote for those, for the 25 reduction, 50 reduction, and 60 reductions? Everything is very even. Let's see if we have one more participant joining. So it's not likely to happen, but these meta-analysis studies reveal that biocontrol interventions reduce crop damage by more than 50%. So this figure already demonstrates the effectiveness in mitigating pest-related losses. So at the moment and at this stage of development in terms of science and all the knowledge that we have an estimate of 50% reductions, which is already quite significant, we have to say. So thank you so much for taking part. So moving on. So we have another question, Rita? Okay, so now it's about the impact on crop yields. So biocontrol on crop yields. So we have crops that are less affected by pests and diseases. So, and again, this is based on a meta-study that, and I can give you some more details. It was a meta-analysis of 99 studies that were performed across 31 different crops. And here, the results showed that biocontrol methods reduce pest abundance and crop damage. So they increase crop yields by over 60%. Okay, 60%. That's the amount of increase in crop yield because we can reduce crop damage and pests and pest prevalence. So 60% crop yields and also crop damage 50%. So we're getting some interesting figures. Thank you so much. And now we have one of our favorite questions. So how big do you think the growth rate for biocontrol market, the global biocontrol market, is going to be in the coming years? And it is an estimate between 2025 and 2033. 7.5%, 12%, 15.8%, or 5%. We know it's growing, like all bioinocula, but you have an idea. And this was a report that was published very recently in January 2025. The project growth, and this is in dollars, is from $9.5 billion in 2025 to $15.5 billion in 2033. So this gives a compound annual growth rate of 7.5%. So it's expecting that yearly this market grows at 70.5%. So we already have some promising, so that we have more to come in terms of biocontrol intervention. So thank you very much for taking part. I think we have warm up. We are well introduced into the topic. And so I will now call to the floor our speakers, which are the stars of this session. So Marisé, I know that you need to leave earlier, so we will start by helping us to understand the regulatory framework for biopesticide. Thank you so much. The floor is now yours.
Okay, hello everybody. I hope you can see my screen.
Yes, all good from our end. Thank you.
Perfect. So again, I'm trying to talk about how we go from innovation to the market. I work in a SynTech research organization, which is the #1 contract research organization that helps companies move from ideas to actually get to the market. We have these values, and we cover the global world. And again, I'll talk a bit today. I'll focus a bit on the regulatory in Europe and the regulatory in Brazil because I think it is relevant. We are located all over the world. And again, each country has its own, I would say, challenges and regulations. I think it's important to mention that when you're going from innovation to the market, you need to have all components of the chain. And even when you have the idea, never lose what you're in, which is the market. So we have laboratories, we have greenhouses, we have field, we have digital technologies, and we have, as I said, our regulatory affairs department. And in order to get to the market, the regulatory affairs, which is at the end a dossier that allows you to have a label, that allows you to be in the market, needs also, I would say, information and studies that come from the lab, that come from the greenhouse, that come from the field. So when you think of how to get to the market, remember that you need all these components. What kind of things do you need? Well, you need, if you have microbiology products, you will need to have microbiology analysis of your products, including the sequence, including possible secondary metabolites, including knowing where it was isolated from. So there's a lot of information. I believe Domenico and Sergio will be talking next week on what you need to know in order to be able to get to the market. Again, if you're thinking of developing a nematicide, you will need to demonstrate the efficacy and the mode of action of your actives and products. We'll go a bit later on what is the difference between active and product. You would also need to make sure, for example, if you're developing either a fungicide or a nematicide, to have trials and studies where you control the conditions under which this happens, meaning inoculated trials. Why? Because when you're working with chemicals, you're kind of shooting, as we say in Spain, shooting flies with cannonballs. When you're working with biocontrol products, you really need to know your environment, and you really need to know the conditions under which those biocontrol products will work, and under which conditions they will not work. An obvious one, for example, Bacillus thuringiensis is a very effective insecticide, but you have to be careful to apply it late in the evening because UV light can destroy the cry proteins that are present there and that are the effective insecticide. So you need to have more knowledge to be able to apply these biocontrol products in your fields. So lessons learned: biocontrol works, but make sure you get all the information to make it work, and also that you know all about the environment to make sure that you use it under the right conditions. Then, as I said, you often, if you don't know a lot about your development, make sure you start early in the lab or in growth chambers before you move to the field. Because in my experience, and I've developed many products that are already in the market, in my experience, when you have an awesome thing in the lab, when you bring it to the field, only between 1 and 10% of those things that in the lab look that are going to be very promises actually are able to perform in the field precisely because these, I would say, environmental component. Again, it's important also to know what is the application method of your product, like whether it's going to be a spray, it's going to be applied in the soil, for which the microbiome, you will hear about it later, is a very important component. So you have different methods of applying your products, and you have to know very well your methods and know how your product is going to be used before you even think of going into the regulatory phase. Because when you go to the regulatory phase, the first question you're going to get is the gap use table. What is the gap use table? Is what is your product doing? Which is your target pest? Which is the application method? What is the concentration of your application? And all that information, typically, you develop first in the lab, then in the growth chamber, and then you move to the field. As I said, the field is also very important. This going from growth chamber to greenhouse to field. And as I said, there's many ways of doing applications that you have to make sure that what is the best for your own products. And again, there's many assessment types. And actually, when we're talking biosolutions, there's two kinds of biosolutions there. The biosolutions that are focused on abiotic stress, nutrition, and so forth. Those are like biostimulants, for example. And then there are the true biocontrol, which you can call biopesticides if they have a killing mode of actions. But many of the biopesticides we currently have in the market are not necessarily killing the pest or killing the fungi. They're actually competing, and there's different mode of actions, which doesn't necessarily mean that kill or you are a pesticide as such. The important thing is that you don't have a disease, you don't have a pest in your crops. Also, it's very important to be aware, as I mentioned, what are your target crops? It's not the same tree crops than row crops like wheat or like corn, or if you're going to specialty crops like ornamentals, which can have more challenges. Also, there are biocontrol products that are working for post-harvest, and that's a whole different regulation and also a whole different way of working. If we are talking, and also when you're developing products, it's important to target in your pest, but never forget that what the farmer wants is to get return of the investment. So you also have to make sure that you don't have, like, for example, phytotoxicity effects, that you recover the yield of your crop. And there's ways of measuring that with digital tools early in the season. And again, in SynTech, we have all these kind of tools to be able to help you. And actually, there's actually robots that are able to allow you to do these in a very high throughput manner. Now, when we're, another important thing is, as I mentioned before, when you are going to put a dossier together, and no matter whether you're in Europe or you're in Colombia or you're in Brazil or you're in the US or Japan, you need to have information on your products on several, I would say, departments. Department number one would be Phys-Chem biological properties. What is the specifications that are defined by your product? What is the concentration of the active substance? Because typically in a product, you will have an active substance and you will have other components that will help it to get to the crop, like adjuvants or surfactants or things like that. So that is, like, say, chapter one. Chapter two is making sure that it's not toxic for humans. So is this toxicology department? Typically, biocontrol products have less toxic properties, but that doesn't mean that being natural or being of natural origin, that you are not toxic. For example, hemlock is natural. You can find it in the field, but it's very toxic. So make sure that you profile your toxicity before going to the field. And then, of course, is ecotoxicity, that is not toxic to, to humans, to non-target arthropods or non-target plants. So that is, friendly with environment. Typically, again, biocontrol products are friendlier with environment. And also that is, we have to follow what happens with the active in the environment. what is called environmental phage, meaning to make sure it has biodegradability components. Again, one main difference between chemicals and biologicals or biocontrol products is that chemicals often have residues, chemical residues, because they are not present in nature, while typically biocontrol products, because if they come from nature, we bring them back to nature, they degrade naturally and you don't have residues. And that's a huge, huge difference. And one of the reasons why when we look at, for example, our own business, Five years ago, bio solutions was 15% of our business. Now it's 60% of our business. And we see innovation going mostly into biocontrol solutions or biological solutions in general. And last but not least, we need to demonstrate the efficacy of our products. We need to do field trials to demonstrate that indeed our products work in the field. If you're in Europe, you have to demonstrate that by pest or disease and crop. If you are in the US, demonstrating by pest is sufficient, unless you're in California, which we also need to demonstrate by crop. But again, moving on, it's important to apply the right regulation for your products. Like if you are going through a biotic stress, there's a European regulation that covers that. If you're going for biopesticides, there's another regulation called 1107, 2009. And be aware, is the same regulation for chemicals and biologicals. So the biologicals have to demonstrate the same things that chemicals need to demonstrate. What is the difference? The difference is that being biological, there are studies that you can use, meaning what we call weight of evidence. If there's literature knowing that a certain microbe, for example, is not toxic to humans, you can already use that information in your regulatory dossiers to avoid having to do the study again. We don't reinvent the wheel. Actually, when you look at the regulation, it's the same regulations for microorganisms, semiochemicals, like for example, pheromones, plant extracts, microbial extracts, botanicals, and chemicals. Regulation is the same. There's actually two sets of requirements. One specific for microorganisms that have recently been developed by the European Union, which is fit for purpose for microorganisms. And again, the rest, whether you have a biochemical, an inorganic matter, or you have a chemical, they all have the same requirements. This is exactly what I was explaining, that microorganisms have a specific requirements different from the rest of the classes. And if you're in Europe, actually this process is very long. Why? Because you have a two-step process. First, you need to approve the active, meaning what is the component that is actually using as a biopesticide. And that is done at the European level, at all 27, there's a European Food Safety Authority that is involved, that is a European agency, and that is approved at EU level. And then you have to have your product registered at member state level, meaning in each of the 27 countries. It's more complex than that because in Europe we have three zones, the southern zone and the central zone and the northern zone. So first you have to do this zonal application and then each of the individual countries. So in Europe is the region where the registration of a biopesticide is more complex. Not only it's complex, but it also takes a long time. It can take, according to the law, it could take as little as 5 years. In reality, in average, it's taking between 7 and 10 years, even for biological products. And that's the reason why in Europe, farmers have a much narrower toolbox compared to other regions. For example, in Brazil, they changed the regulation, and in Brazil, they have almost 500 microbes approved for the use of the farmers. In Europe, we only have 72 microbes approved. And this shows a big difference between both regions. As I said, the process is long and complex. Again, you can check which are the active substances that are approved at European level. And again, that list is publicly available and you can check it. And we've done a recent study in IBMA, the Association for the Biocontrol Products Manufacturers. And actually, when you look at the EU approved active substances, 25% are natural substances, meaning like, for example, botanicals or microbial extracts or biochemicals. 25% are microbes, again, insecticides, fungicides mostly. Another 25% are semiochemicals, mostly the pheromones that can be applied for mating confusion, but can also be applied in attractants that will kill the pest. And then the rest is chemicals. So chemicals are still a big part of the approved substances in Europe. And again, this is, as I was explaining, it takes a long time. There's initiative now in Europe trying to change these and trying to apply for a fast-track approval for biocontrol. It's being presented now by IBMA, and we're expecting to have results by the end of this year, beginning of the next year. This legislation proposal of amending 1107, it will include having a biocontrol definition, because biocontrol doesn't exist legally now in the European Union, and having the possibility of this fast track to kind of, rather than having this two-step process, being able to sell the product once you have the active approved. Because what is the difference between the active and the product? The product has the adjuvants, has the surfactant, has everything on it, and has an additional evaluation. Also for the product, you need to provide all the crop pest targets that you want to have in your label. So it's more complex compared to what you have in just the active. Okay, as I said, process is long, and as I was explaining, once you want to apply to register the active, you have these zones. The northern zone, basically, it's Scandinavian and Baltic countries. Central zone, which are all these, not the UK anymore, and southern zone, Bulgaria, Spain, Greece, France, Italy, Cyprus, Malta, and Portugal. Then, to make it more complex, we have what is called the EPA zones, meaning that you not only need to demonstrate the efficacy in the zone, you also need to demonstrate the efficacy in the different EPO environmental conditions, meaning that, for example, in the southern zone, we have Mediterranean zone in most of the countries, but we also have France, which has a maritime zone, meaning that if you want to register in France, you have to do trials not only in the Mediterranean conditions, but also in the maritime conditions. And when you look at the central zone, you see we have these three EPO regions, meaning that you have to do your efficacy trials covering all these different EPO regions, which again adds an additional layer of complexity when you try to approve biocontrol products. And all this is required by the authorities, and we need to do it to be able to get to the market. Again, as I said, timelines are longer than expected, and process is very complex. When we look at Brazil, we see actually that the Brazilian legislation, even though apparently it's more complex, in reality, it has specific, requirements for alive organisms, and depending on the mode of action, and has these kind of fast tracks for plant extracts, for microorganisms, for biochemicals, and they have a regulation that is in proportion to what the kind of products you have. And what we're aiming is that Europe will learn from Brazil, recognizing that biocontrol products have specific characteristics that should allow for faster evaluation without jeopardizing the safety of humans and the environment. And again, you also have different agencies involved in Brazil. And with that, I believe I'm done with my 15 minutes, and I'll be happy to take questions.
Marisé, I'm sure that you have your timer there, because you're spot on the 15 minutes. So well done. Thank you so much. Great overview. I think a lot has been left to say, but I think we have a good understanding. So you mentioned that for a biocontrol product to be approved, there is a rigorous path and a very long one to ensure that the product can come to the market. Today we were with a group of farmers, and we heard a lot of complaints about how trustable these products are and that they don't work, and we're not confident that we have the right substances as they claim they have. We're not confident that we'll have the right efficacy. In terms of, having a product that has gone through that process, what could you say to these farmers?
I can say if they're in Europe, the efficacy has been proven and has been proven, I would say, under their own conditions. What is critical is that they know how to use the product. As I said, farmers of biological products need to be more educated than farmers that are using chemicals. Chemicals, again, you shoot to everything, you kill everything, and then it works. But it's not sustainable in time. And we have a lot of examples where with chemicals you have resistant fungi, you have resistant insects. Again, I can tell you as an example, I was in Almeria in Spain last week. In Almeria, again, very thriving region, they only use biocontrol. Why? Because their markets are export. They cannot have any residue in their crops. So they had to learn how to grow without any chemical input. And Almeria, the whole region, doesn't use chemical inputs. They just use biocontrol. What happens? That the level of knowledge in the farmers has raised. And their markets, meaning export market, having very high, I would say, quality standards, has driven them to actually learn how to use biologicals. So I can tell you, if there's a biological product in the European market, it works. What is important is make sure you talk with your technical advisor and you make sure to use the product as is recommended when it's recommended. Because what I see is often farmers use without the knowledge. A very good example. If you have a microbial product, farmer might be tempted to use more quantity than is recommended in the label. Of course, with a chemical product, if you use 10 times more, you feel that you're going to kill 10 times more. With a microbial, if you use 10 times more, what happens is it will work much worse. Why? Because you will have intra-species competition. If you have too many microbes, they are too close together, they don't have room to grow, and then you will have no protection. And you'll say, oh, I used 10 times more microbe, it didn't work. Of course it didn't work, because you're not following the label requirements. In order for it to work, you have to apply the right dose. And if you apply 10 times more, it will not work. And if you have these chemical mindsets that 10 times more is going to be better, then of course you're wrongly using the product, and then you say microbes don't work. It's not that microbes don't work. Basically, you didn't read the label, my dear. So this is one of the many examples I found in the field where farmers have this belief that biologicals don't work, and it turned out that more often than not, they have not read the label and they have not used the label. Another example, if you are using biocontrol and you are using biostimulants, you have to make sure that there's compatibility, because if you're using biostimulants with a lot of microbes in them, and then you try to apply your biocontrol microbe, then your biostimulant microbes will not allow your biocontrol microbe colonize your soil. And therefore, of course, it doesn't work because you already had put many other microbes. So you have to understand, and you have to know more agronomy, you have to know more physiology, and especially more ecology and more biology to be successful. But I can tell you that farmers that are able to change this mindset from chemical without, just shooting to the blind, versus really trying to understand their crop and trying to help their crop, at the end, it's more sustainable, less resistance, we've done a lot of economic analysis, and actually the return of the investment is much better, but you have to apply and use them with science and with knowledge.
To apply them correctly. And I think you
Exactly.
brought a very important point, which is knowledge and understanding of what we are dealing. But I think a stakeholder that is also very important here, and so that not everything relies on the farmer. It's also these commercial teams that are out there trying to
Absolutely.
assist, and you know, the role that they also play in terms of transferring that knowledge, and sometimes they don't have also the knowledge because they were just selling
No, no,
chemicals, and all of a sudden they are selling, you know, biocontrol
exactly.
products. Okay.
Exactly, and we've seen that a lot. Companies that come from the chemical background, and all of a sudden they have biological products in their portfolio, but they've never worked themselves with biologicals. They give roaming extractions. So it's not only farmers; it's also, you're right, advisors that don't know their own products because they come from a different mindset, a different background, and they make, I would say, wrong recommendations as well. So knowledge is very important all the way through the chain. I fully agree.
Marisé, just one final question, since you are here, and I know that you need to leave earlier. You mentioned the lengthy time that these products take to come to the market, and we also understand that the more products, the more trials, the more access that farmers have to the learning, the learning curve, the faster the learning curve, so the more we can improve in terms of using this type of intervention. So what do you think are the regulatory steps that we could take to accelerate the approval of this low risk biocontrol products, but of course still maintaining safety and efficacy standards? So how can we shorten this time to market?
Well, I see three things. First, to support, biocontrol industry, claiming that EU farmers are in disadvantage compared to the rest of the world. And we have agricultural products coming to Europe where farmers have a very huge toolkit, when here they have a very narrow toolkit. So farmers have to complain, not try to keep the chemicals. Please ask for more tools because the tools are available to other geographies. Make sure that you have those tools available. Two, when there's the option of exceptional authorizations, use these exceptional authorizations not only to bring back the metal sodium and the candidates for substitution and all these kind of very toxic chemicals. Use these exceptional authorizations, Article 53 of the 1107, also for new biocontrol tools that have already proven their safety in other geographies. Try to get early access to as many tools as you can. And third, as I said, by the end of the year, we are hoping to have these initiative legislation to amend 1107. So you talk to your members of parliament or the members of the EU Parliament, to your country representatives, that they actually support innovation, so the farmers can have access to the full tool. I mean, innovation will bring competition, will bring more help in the field. So I would say these would be my three recommendations. And again, I'm really sorry I need to leave, but if any of the people in the room have additional questions, my email is available through the Food4Sustainability, and I will be delighted to answer additional questions, because again, 15 minutes is just a screenshot.
Teaser. Yes, Marisé, if you can
teaser.
send us your presentation, we will be very thankful, and thanks for your participation here in our webinars. All the best, and now we will move on. Joana, I will call Joana to the floor. She's the Director of Soil Health and Function Laboratory. Joana, thank you so much for being here. So the floor is yours, ready whenever you are.
Thank you, Claudia. Hi, everyone. I'll just share the presentation. Please let me know if you can see it correctly.
Yes, all good from this end.
Okay.
Thank you.
So, hi, everyone. As Claudia said, I'm the head of the Soil Health and Function Lab at Food4Sustainability. Today, I'm going to be talking to you briefly about our lab and about how what we do in the lab can help in this transition from to biopesticides. So, very Okay, so very quickly, introducing the lab. The lab is located in Portugal, close to Lisbon. The lab was inaugurated in 2023. It is a molecular biology lab at this moment, state-of-the-art lab. So we have automated extraction of DNA and RNA. We have endpoint PCR, and we have an Illumina platform for conducting the soil microbiome analysis through next-generation sequencing. We started our operations in May of last year, and until now we have already performed over 1,200 assays in both client and project soil samples. Most of our samples at this moment are from projects, R&D projects that we have at Food4Sustainability, but we are seeing a very good acceptance of this service and this technology from producers, which is for us a very important important sign. Like I was saying, as available services at this moment, we can perform the soil microbiome analysis. We are focusing right now on bacterial and fungal communities, and we are also able to perform through a partner lab this soil physical chemical analysis. During this year, we will be expanding our portfolio of services by a lot. We will be including the analysis of soil enzyme activity, microbial enzyme activity, as a complementary tool for assessing the microbiome functions. We will also be able to include the detection of organisms by PCR and real-time PCR. In addition to the bacterial and fungal communities, we are also including nematode as a target for our soil microbiome analysis. And we will also be moving a bit from soil to other important matrices for agricultural purposes, like water and plants themselves. And in addition to combine and integrate all of this, we will be expanding our facilities to include capacity in classical microbiology, which is also something that our clients ask for on a daily basis. Okay, so today I would like to start with this slide. So what are the questions that we need to answer for transitioning to biopesticides? And I want you to keep in mind this slide because I will be returning to it in the end to wrap up. So what are the questions that we need to answer? First, we need to understand what is the microbial diversity. So what organisms are already present in the soil and how are they interacting with plants and with among each other, like Marisé was saying, is also a very important point. Then what are the soil functions how are the soil functions going to react to the biopesticides that we'll be implementing? How can we also achieve biopesticides that are robust in different conditions? In terms of nutrition, we can also ask how will the biopesticides impact the nutrient cycling? And in terms of health, which is the most important question, for last, are the biopesticides effective against the target pathogen? So to answer these questions, our tool of choice is the microbiome analysis by NGS. And I am going to just quickly give an overview of how we go from the sample to the insights that we are aiming. So first, we start with sample collection, then we proceed to DNA extraction and sequencing, moving on to data analysis, and then finalizing with result interpretation. For the sample collection, we should be aware of the experimental design. So how do we select the samples? What are the sites of sampling? what are the moments of sampling, and what is the number of samples. This is also something that we need to, like Claudia and Marisé were mentioning, we also need to educate farmers on this, because just doing one sample and expecting to have all the answers is not reasonable, and so we need to include a detailed experimental design and an appropriate number of samples, making sure that the samples are representative from the type of soils that we have. Also, if we are evaluating efficacy, we should also include a baseline before application, and then samples after application to be able to monitor that efficacy. In regards to the sampling protocol, we should also collect the soil at a defined depth that is appropriate to what we are targeting, and making sure that we send the samples to the lab in a properly fit package, properly identified, with geotagging if possible, and under controlled conditions at all time, especially for temperature. When the samples come in to the lab, what we do as soon as they get to the lab, to avoid any degradation and any further impact of inhibitors, we move on to genomic DNA extraction. And in this case, we include a mechanical and chemical cell lysis, because soil samples, as you know, contain hard-to-lyse microbes, and so this combined method of lysis is is critical for the success of the downstream applications. Also, as the removal of inhibitors. So, as you know, soils are rich in humic acids, which are naturally inhibitors of PCR reactions. So they are going to, if not removed, they are going to inhibit any downstream application as well. Then finally, to make sure that all the process was done correctly and we have good DNA, we should also do a quality control. So checking of the concentration, the purity, and the integrity of any DNA before moving forward. So for DNA sequencing, I'm going to briefly explain the process. This is the process based on an Illumina platform, so short-read sequencing and sequencing by synthesis. There are other methods for evaluating microbial communities of the soil, but for now, this is what I'm going to focus on. So first, with the extracted DNA, we must do a PCR amplification that targets conserved regions, which are flanking variable and informative regions. So in this way, we target a region that is common to all the microbes. For bacteria, we have the example here in the picture. We have the example of the 16S ribosomal RNA gene, which is usually the most common one for identifying bacteria. And we target conserved regions, which are in blue, okay, that are flanking the variable regions that are in pink. The variable regions are the regions that we can compare between them and compare against reference databases to identify the species. After PCR, what we do is the preparation of the sequencing libraries. And basically what we do is we take the amplified fragments resulting from the previous PCR. We do adapter ligation and indexing of the samples of the libraries. Sorry, we do normalization and purification of these libraries. After that, the libraries are pulled and are loaded into a flow cell. So the flow cell is the chip where the sequencing happens. And the fragments are hybridized on the flow cell surface. And they are hybridized through the ligation of these adapters, which are complementary to adapters that exist on the surface of the flow cell. After this loading, what happens is another amplification of the fragments that are bound to the flow cell, which generates clusters of sequences. Then the sequences are produced, complementary sequences are produced. So this is a sequence by synthesis. That means that we are going to produce new sequences complementary to the templates that are bound to the flow cell. And each time a nucleotide is incorporated, a fluorescent signal is emitted, and different colors will mean different nucleotides. And that color signal is then translated into a digital signal that is recorded on a file, okay, on a text file. And so basically this detection of fluorescent nucleotides is used to generate the millions of reads in parallel that we see in sequence by synthesis. On the final part of this process, and after the data is generated, so we have millions of reads in one run, in one sequencing run, and we have several thousand reads per sample. Okay, so the first thing that we need to do to digest all that data is initial processing, and this initial processing comprises the trimming and the filtering of sequences based on quality. So we only keep the high quality reads. So anything that is low quality for any reason is discarded, and we keep high quality reads. And that is the way that we make sure that the results are reliable and that we can trust any identification that comes out of that data. So these sequences are then clustered, which means that they are grouped in sets of similar sequences. And those similar sequences are then matched against reference databases to achieve a taxonomic alignment. And that taxonomic alignment then tells us that that group of sequences belongs to that species, okay? In the end, we will be left with a list of identifications, with a list of all the species that are present in that microbial community. Okay, so going back to the first slide, we can see that through sequencing, we are able to understand we are able to answer all the questions that I placed on the beginning, on the first slide. So we can assess the microbial community based on the taxonomy. We can also quantify the ecological functions that are performed by soil microbes in that sample. We can also evaluate the ability of the communities or populations to remain unchanged when stressed. So we're talking about resilience, we're talking about functional redundancy. On the nutrition side, we can also assess what is the nutritional status of the soil based on the potential microbial mobilization of macro and micronutrients. And in terms of health, we can also assess crop health according to the detected pathogens, especially if we can support this information with microbial indicators of biocontrol, hormone production, and stress adaptation. So we go full circle on all of these indicators of what is the general status of the soil when we look into the microbial communities. And at the end of this, at the back end of this, what do we use this data for? So we use these insights to provide recommendations to the producers. So we integrate this data with data from other analysis. We are talking about physical chemical analysis, enzyme activity analysis. We also integrate data from climate conditions and plant development, and we recommend agricultural practices such as crop rotation, mulching, incorporation of other microorganisms, and similar sustainable practices. So that's basically what I had to present today. I'm happy to take any questions that you may have.
Thank you so much, Joana. Again, spot on timing. Thank you so much. It looks like British punctuality, Mojgan. We are, learning with you. Fantastic. Joana, thank you so much for such a thorough and detailed explanation about what soil microbial analysis is. One question. If a farmer requests this type of analysis, what can they do with the results? Are they able to interpret the results? How can they deal with this information?
Well, it depends on the knowledge of each producer. We have producers that have more knowledge already on this topic of microbiome, but simply providing a species list with a relative abundance is not going to be enough for most farmers, for most producers. So I think that it is important also for farmers to have access to consulting, access to training, to be able to understand what exactly does this mean, okay? And how can this list of species be translated into these indicators that we provide?
Okay, thank you. So this is not very easy to look at the data and draw conclusions. So you need more information, you need other analysis, you need to understand the context, the practices that the farmer has. So this is almost like a collaboration work between
yes. It is a very close. It is a very close work with the farmers, where we try to collect the most information possible at the moment of sampling, ideally. Sometimes farmers don't have that full set of informations that are essential to understanding the results. Yeah, but yes, this is a very one-on-one work that we do also with the farmers, to make sure that we understand their context and what exactly they are also aiming.
Okay, fantastic. Thank you so much, Joana. We have a question from Teresa. Hello, Teresa. It's nice to see you here. Hope you're well. I can see that you're still following our work. Great to see you. And Teresa wants to know what are the function analyses that we normally do, and what are the softwares that we use. So in terms of functional analysis and the types of softwares that we use. Joana, can you answer?
That is a very interesting question. So we have our own soil microbe database where we have a list of 35,000 entries already, where we have this functional characterization of microbes. So based on the species list, we can infer the functionalities that are happening at the moment based on the genome content as well.
And this is all scientific evidence in terms of the relationships between...
In official documents, yes.
Okay. Thank you so much. Joana, thank you for your contribution. If you have more questions, you can just pop them here in the chat. I will move on with our session. We still have Mojgan, but Joana is going to be here, so she's happy to answer all the questions. So Mojgan, good afternoon. I know you also have a very exciting and brand new topic for us. So let's hear about Phage, what they are and what they can do for agriculture. The floor is now yours.
Perfect. Hello, everyone. Thank you for having me. I just put my yes, my name is Mojgan Rabiey. I'm from University of Warwick. And in my research group, we work on using bacteriophage or phage to control important crop diseases. So we work on different pathosystems One of the systems that we work on is the black rot of brassicas. So we all know about brassicas, so like kale, cabbage, broccoli, cauliflowers, and so on. And vegetable brassicas are the third most important crops in the UK, and UK produces around 300,000 tons of them, worth nearly 400 million pounds. But these brassicas are susceptible to black rot that is caused by a bacterial pathogen called Xanthomonas campestri. And you could see the symptoms on the leaves and how much it could impact the crop yield. So it's quite devastating for the farmers and growers. And currently, there is no control for the disease. For example, we can't breed for resistant, or we can't use chemicals, because also this pathogen is quite complex. We have over 11 races of Xanthomonas campestri, and each race has different pathogens and each different strains, and so it's very complex disease. So in my group, we try to use phage to control and treat the disease. Another system that we also work on is bacterial canker of cherry. So again, it's a very complex disease, but it's caused by Pseudomonas stringi, a bacterial pathogen that could infect over 200 different plant species. And the pathovars of Pseudomonas stringi is Pathovar stringi and Pathovar murospronorum, race 1 and race 2. From now on, I call them PSS and PSM. And if you look at this, for example, phylogeny tree here, you could see that PSS and PSMs belong to different phylogroups. It means that they have different pathogenicity and virulence, and each phylogroup has different strains. Again, very complex pathogen. So this pathogen could infect all parts of the plant. So it could cause shot hole on the leaves. It could damage and cause necrosis on the fruit, and it cause huge amount of oozing and bleeding on the trunk of the tree. That it seems that trees bleed to death. And currently we have no control. For example, we used to use copper, but because of the legislation and because also we have bacterial resistance emerging to copper, we can't use that anymore. Also, we can't breed for resistance because this disease is very complex. There are different pathogens with different pathogenicity and virulence, so it's not possible. So again, we try to use bacteriophage as an alternative. So bacteriophage or phage are viruses that infect and kill bacteria. And how do they do this? They bind to the bacterial cell surface here. They inject their genomic material inside the bacterial cells. They hijack the replication machinery. They make their own particles, and they lyse and burst the cells open. And each of these bacteriophage, then they go and infect the neighboring cells. So this is the beauty of using phage. This is a live interaction going. So it's not like antibiotics that you use once and bacteria resistant emerge, but this is a live interaction. One phage infects one bacteria cells, at the same time produces hundreds of other phages that they could infect the neighboring cells. And these phages are very host specific. So it means if this phage could only infect the Pseudomonas stringi, Pathovar stringi, it means that it cannot infect other Pseudomonas or other bacterial pathogens. So that's good, because then in terms of safety, we want to use the product that are very specific to the pathogen that we work on. So it increases the efficacy. Also they don't impact the other microbes in the environment. So for this reason, we went to different cherry orchards around the UK. We collected 70 phages and that they could infect PSS and PSM, and we collected them from bark, soil, leaf, and so on. And you could see the plaque morphology in the slide here. So the first thing we do to understand the safety of phages is we test them against a range of different pathogens. So, for example, here you could see MR1 to MR18 are the phages, And on the x-axis, you could see PSS, different strains, PSM1 and PSM2, different strains, and you could see other pathogenic Pseudomonas and other beneficial microbes, those microbes that are we like to have them in the environment and they benefit the plants. So in yellow, you could see that phages could infect, and in purple, they cannot. So you could see that none of the phages could infect other pathogens, and they cannot infect any of the beneficial microbes like Pseudomonas fluorescens. So in terms of safety, we first test this to understand, okay, they are safe to be used. We could see the host range, we could see how many different strains of PSS and PSM they could target. Next is to genome sequence them. So we genome sequence phages because we want to understand if they carry any toxin genes, any virulence genes, or any antimicrobial resistant gene. And none of the phages that we had carry any of the toxins and antimicrobial resistance. And based on the genome sequence, we found that phages, some of them are unique for the first time we find them. And based on this, the phages belong to 5 different groups. And from each group, then we chose one phage and we further characterize them that I'm going to focus on for the rest of the presentation. One of the first thing that we also try to understand when we characterize our phages is in terms of if we want to store them or if we want to apply them in different conditions, in what temperature they could survive. So, for example, if we want to store them for a long time, or if we want also for the application in the field. So here, what I'm showing you, we counted the number of phages, and here you could see the PFU or plaque forming unit. So the number of phages that we counted. And here on the x-axis, there are different temperatures. For example, -24, 20 degree, 27 degree, and 37. And for example, this MR4 phage could survive up to 6 months at different temperatures. Same for MR6 phage. So it shows that in terms of the viability and infectivity, they could survive at a range of different temperatures. Next, we test the efficacy of phages to understand how quickly and how effectively they could reduce the bacteria population. And how we do this, we measure the optical density of a bacterial population, for example, PSS, in the absence of phage and in the presence of phage. For example, you could see when we added phage ML6, you could see how quickly this phage ML6 reduced the bacteria population. So these ML phages are all very effective in vitro or in the lab when we test them in the presence and absence of the pathogen. So now we know that phages are very effective in the lab. And so what about in the plant environment? So we do a lot of glasshouse tested. And so to understand, okay, those tests that I showed you, they were in the lab, but what about the real world environment? So that's the glasshouse. So for example, in terms of cherry trees, we infect the woody tissues with the pathogen and with phages, whether individually or in the mixture of 5 phages. And we measure we look at the symptoms, we score the disease, and we also isolate the bacterial population. Again, we count the numbers to see whether the application of phage reduced the bacterial population or not. So here, for example, look at the symptoms. Here, when we have the pathogen, Pseudomonas stringypathoba, you could see the browning lesion on the woody tissues. If we apply MR6, so the phage MR6, you could see the reduced symptoms compared to PSS. And look, when we apply the mixture or cocktail of 5 phages, you could see almost no symptoms compared to negative. So, and also if we measure the bacterial population after we infect them with phage, you could see that it could reduce the population significantly. So all these results show that yes, so the phage application in vitro and in the glasshouse could reduce the bacterial population significantly. And when the reason we use mixture or cocktail of phages is because as I said, look, when I showed you the video, each bacteriophage, when attached to the bacterial cell surface, they use different, what we call it, receptors. And based on what receptors each phage uses, if we mix different phages that they use different receptors, we could basically target different strains or different isolate of the pathogen. So that's beneficial to use the cocktail. Okay, so now we moved on to the field trial. So this was the first ever field trial of phage in the UK. So we went to the field but not knowing what to expect, because all the experiments we do, they are in the lab or in the glass house. But when we go to the field, we have different temperatures, we have different humidity, we have UV intensity, we have different microbiome. But so what we went to the field and said, okay, for how long, for example, these phages could survive, and does phage resistance emerge to the bacterial population in compared to antibiotics, for example, if you use copper, and we wanted to understand if phages, for example, impact other microbes. So from our lab experiments, we know that they wouldn't, but we need to know in the real world environment. So we went to the cherry orchard, and we selected our trees, and we collected our baseline samples to test whether we have native Pseudomonas stringi, Pathos stringi, or native phages. And when we were sure there were no native phages or bacteria, we then sprayed the leaf and shoot in the field with a pathogen, and with phage, or with pathogen and phage, and or a buffer as control, just to compare. So these are our treatments. So control, we have a cocktail of 5 phages, we have pathogen PSS, and we have PSS and the cocktail of 5 phages. So we have all the possible combinations, and then we collected samples every day for 4 days, and then after 30 days, and we looked for phage and bacteria in all our samples, whether we applied them or not, just to make sure there's no cross-contamination, and then so on and so forth. So what I'm showing you here is the phage count, or plaque forming unit. So phage count here, you could see the treatments on the x-axis, and you could see the different time points. So at the baseline, so if you look here, we don't have any natural phages. So that's good, because then we were sure that the phages that we applied, these are our phages, not what are already in the nature. And then if you look at the cocktail of 5 phages, you could see that after 3 days, we can't detect phages anymore. But if we have the pathogen and the phage, we could see that phages survive up to 30 days. So what it means, that it means that in terms of safety, so for example, if you go to the farmer and say that, oh, we want to apply phage, the first question they ask is like, how long phages could survive in the field? So phages survive as long as the pathogen is there. And the reason is that because phages use the bacterial population to replicate. And the reason we use biocontrol is to reduce the bacterial population below the threshold that they don't cause any more damage, not to eradicate the pathogen, because it's part of the microbial community. So this shows the safety of phages. Okay, only when the pathogen is there, they could survive and replicate and reduce the bacterial population. So the next question that we wanted to answer was whether the resistant emerge in the bacterial population, because that would be a big problem, because we don't want what happens when we apply antibiotics or coppers and so on. So what we did, and so as I said, when we applied the pathogen in to the field, we re-isolated those PSS from the field, and we used the wild type phages again to see if they could kill the bacterial population that we re-isolated from the field experiment. If the wild type phages could kill those, it means that resistance doesn't emerge. But if the wild type phages could not kill them, it means that the bacterial population evolved and made the resistant emerge. So you could see here that in red and green, these are the bacterial population that we re-isolated from the field after we applied them. And you could see that wild-type phages could reduce the bacteria population significantly compared to the control. So it means that no resistant image in the bacteria population. And we also backed this up by sequencing them and finding that there was no mutations in the bacteria population. So all shows that the application of phage does not lead to emergence of resistance in the bacteria population. So it means that phages are safe to be used. Next, also to understand the safety of the phages, we look at the growth of the bacteria population. We don't want to create for example, if you apply phages, we don't want to create a bug or the pathogen that is, they could grow faster or they could grow differently from the wild type phages, bacteria. So here again, you could see it in red. That these are the bacteria population that we re-isolated from the field, and compared to the control, they grow at the same rate. So the application of phage did not impact the growth of the bacteria population, so it doesn't impact the fitness. That's again confirmed the safety of using phages. Next was to understand in terms of safety, could the phages impact the microbial community? Joana touched upon this. So Joana talked about the microbial community. So in terms of bacteria, for example, we do 60ness. That is the region that we sequence in for bacteria population. And here I'm showing you the alpha diversity or the diversity of microbes within the community. And day 3 and day 30 in red. And you could see that the time has most impact on the microbial community and not phages. We did a statistical analysis and we found that phages did not impact the bacterial community. Same for the fungal community that we do ITS sequencing. So again, time has the most impact on the bacterial fungal community, and phage didn't have any significant impact on both fungal and bacteria. So again, in terms of safety, it shows that phages are safe and they don't impact because they are very specific, only impact the bacteria population. So we answered the question that we wanted. So phages could survive as long as the host is present, the pathogen is present, and they don't impact the microbial community and no phage resistant image in the bacteria population. So it was a very successful field trial for us. You could see that the amount of plates, for example, we used in 1 day, and we made this phage picture here. But it's very labor intensive work, but all in all, it helped us to understand the application of phage in the field. Just to have the final conclusion, here is how do we apply phages in the field? One of the, for example, we could use the phage as to cure the disease. For example, in terms of cherry canker, if we see the symptoms on the trunk of the tree or on the woody tissues, we could apply that directly to the symptoms. Or we could use it as a systemic measure. For example, it could go up and down. We could use it as a preventative measure. We could, for example, what we did as a spray method, we spray to the field. If we know that, for example, that field gonna suffer from the pathogen, you could use it as an irrigation method. So you could put it as to water the plants, or we could use it as a seed coating and apply that in to protect the seeds from getting infection. So there are different methods, but for example, for farmers, we know that a spray method, whether to the lesions or into the depends on what time of the year we're going to use, or depending again on the pathogen, on the disease, the application could be different. So in that note, I want to thank everyone that has been involved in the project and all the funders, and thank you all for listening, and I'm happy to answer any questions.
Mojgan, thank you so much. This was a very promising presentation. I'm glad that I know that you come from university because you look like a commercial company. It's all wonders. It's all wonders. So very inspiring, and above all, it also gives hope to this transition, and to especially to farmers that need to deal with diseases and pests in a regular occasions, and now we can at least have some hopes of new interventions. Let me just point to our participants that we don't open the conversation. You will need to use the chat. Mojgan, one question. Stefan asks here, I think you understand that, you answered that during your presentation, but can bacteria become immune and resist to constant use of the same phage?
Okay, so that's a good question. So as I said, the beauty of using phage is that there's a live interaction going on between phages and bacteria, because like phages bind to the bacteria and need the bacteria to replicate. So if they bacteria, for example, resist, if like or evolve to immune against phage, bacteriophage also evolve, right, to find a way to infect again. So for example, in the video I showed, as I said, the phage bind to the receptors. If bacteria are going to say, okay, I'm going to have a mutation in my receptors, so phages cannot bind to the receptor anymore, phages say, okay, I'm going to evolve to again adapt to that mutation. So there's like a co-evolution happening. So we call it like an arms race between phages and bacteria. Even if resistant image, if the mutation have resist, phages also evolve to resist. So, but this is what we see in the lab, but in the real world environment, in the plant environment, resistance doesn't emerge as often. This is the reason is the bacteria needs to, there's a trade-off. Bacteria could resist and lose its, because as resistant emerge in the bacterial population, they lose their pathogenicity and virulence. So bacteria needs to think, okay, should I resist to phage and lose my pathogenicity so that I can't infect and survive? Or should I just infect and survive and let the phage do its work? So there is a trade. So there is the emergence of resistance in bacterial population in the leaf environment or in the plant environment is highly unlikely to phage because of the trade-off between being resistant to phage or being pathogenic and survive and do the infection. But again, as even if the resistant bacteriophages, again, they could evolve to overcome that resistance. So it's not like antibiotics that you use them and say, okay, that's resistant image, that's it, we can't do. But phages, because of the live interaction, there's always a phage that could infect again, even if the immunity happens in the bacterial population.
Thanks for that explanation. It was very clear and fascinating how nature works. Stefan also wants to know, how does application of phage on on plants is going on when we have two bacterial infestations on the same plant? And I think this is also common for several diseases that are a combination of microbes.
So that's why I mentioned we use a phage cocktail or mixture of different phages. So for example, if we have Pseudomonas stringyxi that infect, or then we have another pathogen that also infect, we could mix our phages together or make a cocktail or mixture, and then we could apply them. And the reason is because then each phage gonna infect a specific pathogen. For example, in our case, we have the cocktail that we make could infect PSS, could infect PSM race 1, it could infect PSM race 2. Then the mixture could target more pathogens, more pathovars of the same pathogen, or more strains of the same pathogen. That's the reason we could. And so this is usually we prepare cocktail. It could be up to how many phages that we want. We usually test this phage cocktail before we apply them. We need to make sure that they don't use the same receptors, they don't antagonize each other when we put them in a mixture. And if they target different receptors on the bacteria, then they're unlikely to antagonize each other or impact each other. That's why the phage cocktail and mixture comes into place in that case if you have more than one pathogen.
Oh, sure. One question. In terms of science, how advanced are we in knowing exactly what is the phage for a specific bacteria? So, for example, I can give an example, a very specific bacteria that is causing disease here in Portugal in certain crops. Can you quickly say, oh, but we have the phage for that bacteria, or no, we still need to research, to look for the phage, see how it works? You know, what is the stage of science in this sense?
So phages are the most abundant microorganism on Earth. So basically they're everywhere. That's why that's good to use. And then so we usually what we do, if we know, we usually have like a library of phages in our phage collection. So we know that, for example, what phage impact what, but at the same time, we know that there are new pathogens emerging every time. We know that, for example, with climate change, there's movement of pathogens, so we find new pathogens everywhere. But we have like a protocol in place that we use depending on the pathogen, whether it is in the soil, whether it's in the leaf, whether it's in the bark or woody tissues. We collect the samples from the infected area, and we could isolate new phages. And as I said, because they are everywhere, we are likely to find them. And then we usually characterize them in the lab, as I explained, a step by step, and how do we characterize them. But usually the first step we do is the host range. So we know what pathogens we want to use phages against, and then we do the host range, and we screen our phages against the pathogen to understand what they could target and what they can't target. And then the rest comes with the characterization and so on. So we have our like enhanced protocol in place, and that we know from the isolation to characterization to application. And usually like depending on the pathogen and depending on the disease, we could then come up with a phage cocktail or even like a single phage that we could use against different pathogens.
Okay, great. Because one of the questions that we have here in the chat is whether regulations allow phages in the field. So are these commercial products? Are they available already or not yet?
Right, not yet. So, for example, in the UK, legislations are very tight. We need to prove to the government that phages are safe to be used. That's why we need more data, more field work trial. And so for example, the field trial that I showed you, the result, we had the license to use them. But in the US, we have phage products that are commercially available, but because the US legislations are different from the rest of us in Europe and UK, but there is a light at the end of the tunnel. We are getting closer to that light, and hopefully soon we have phages that we could use to treat different pathogens.
Okay, good. So we look forward to that. We're going to be on the lookout. We had another question here in the chat. So phages, they die without the host. Is that a limiting factor for using phages?
I think that's a safety measure, because like we don't want just for phages to impact to be there or impact because like, as I said, the whole purpose of biocontrol is to reduce the bacterial population below the threshold that they don't cause any damage. But we don't want to eradicate the pathogen, because this is part of the microbial community. We don't want just to impact the microbial community as it is, because it is how, for example, if you look at the microbiome in our body, in our gut, we don't want just to interfere and change them all the time. We want, unless we have a disease or we have infection, we don't want to take antibiotics, right? So again, it's the same for phages. We don't want just to apply phages or phages to be there to impact the microbial community. We want to only use it if we know that there's going to be a pathogen that is going to cause disease and damage. And that's why. So again, it's just how to keep the balance of the microbiome within, because the plant microbiome is very important for its growth and its productivity. So that's why we don't want phages just to linger around. So this is the safety measure rather than a limiting factor.
Okay. Thank you for your explanations. So what are the sources of phages? Are they epiphytic? Can you clarify?
So it depends on the pathogen. Everything depends on the pathogen. So, for example, in terms of Pseudomonas stringy, we have Pseudomonas that is epiphytic, and we have those that are endophytic. But epiphytic means that they live on the surface of the plant, and endophytic they go inside and they go inside the cells. So again, depends, pathogen to pathogen is different. Some pathogen remains under like epiphytically, and phages could interact. So again, so this is also the area of work that we still need to know more information. This is the what we are doing currently in the lab, trying to understand where phages go, whether they stay localized, whether they go systemic, and what happens when we apply them to the plant. But again, the pathogen to pathogen, pathosystem to pathosystem is different.
Okay, interesting. One final question. Does Phage application needs to be repeated in time, or is it one-off?
Again, so the disease to disease, pathogen to pathogen is different. For example, in terms of cherry canker that I just explained, we know that the pathogen is around all year round. So we know that it could infect the growing season start, it could infect the leaf, it could then infect the flowers, it could infect the fruit, it could then depends on if we want to use them just as a preventative measure. So we don't want them to have it at all. So we could have just one application at the start of the growing season. But if we know that we have a field that from last year result there is like the pathogen is like severe, so then we could have multiple applications, for example, once a month, because depends on how long phages could survive. So for example, in that terms, we know that phages survive up to 1 month, so the application once a month would be enough. And then again, if it's like for the brassicas, then again the application is different. For brassicas, we usually, for example, could apply it as a seed coating, so that would then protect this from getting infection during the growing season, or we could then use them as a spray method as we transfer the seedlings from glasshouse to the field. This is how the sowing happens. So again, from the different plants to different pathogen is different, but depends on what application we want, whether it's preventative or whether we want to cure the disease, whether we want to reduce the bacterial population. The application would be different, whether to apply regularly, or this is again with what happens with antibiotics. They apply them as they need it. So this is again happened with the phages.
Very interesting, Mojgan. I can just give here, the opportunity to conduct some field trials here in Portugal. Food4Sustainability has some programs like the test beds where we try and pilot some of the technologies. So anything that we could accelerate this knowledge and to see how it can be put at the service of farmers, because pests and diseases essentially are a huge problem, also in horticulture. So if you need some testing fields, and I'm sure some of our participants will also be keen to know a little bit more and to give it a try in these phages. It's very, very interesting. So I'm going just to go for one final question. In the meanwhile, for all the participants that wish to have a certificate, I just left here the QR code, or I think it's also here in the chat. You can fill out this form while I'll just take one final question to Mojgan and to Joana. So, and the question is, if you had a lot of resources and you are really, really trying to push forward this transition from pesticides to biopesticides, what would be, the big breakthrough that you would try to push forward to make sure that it would happen, this transition?
Well, yeah, so I think if I had all the money in the world, like so basically, all my research career I worked on biocontrol, whether it was phage or whether, for example, I use beneficial bacteria, or I worked on endophytic fungi. So these are the future. We've seen use of antibiotics in humans, that how the antibiotic resistance emerging and causing a lot of damage to human system, but also in agriculture, how devastating it could be. And we know that again with climate change and how bacterial or the pathogen gonna a new pathogen gonna emerge. So again, so use of natural microorganism is the way to go. And the biocontrol is my passion, and the phage are now my life. So I would say that I would spend all this money on this phage production, because, as I said, these are everywhere. So why not to use them when we can?
Okay. Thank you. Let's see if more comes into your research lab to make that dream come true. Joana, your thoughts on this?
My thoughts on this is that's a very interesting question, actually. I would say that if I had all the resources, I would dedicate myself to pressuring policymakers into making this kind of technology more available to the general public. So making these analysis and these field trials mandatory and more accessible. I think it would mean a lot for the sorry, I'm missing the
farmers,
word. For the
the
farmers
farmers
and for
to
advancing in this field as well.
Okay, so we are all here in the same boat. Okay, thank you so much for being here. I will now have to call to the end this great webinar. I hope that you enjoyed. I think we had a great kickoff to our series. Just quickly to remind you that next Thursday we will continue, and next Thursday we will look at specific microorganisms in pest management. So you are all invited to come to join our discussions, and as we said, we want this to bring you also new ideas on how to approach your work and your field of expertise in everyday life, hopefully emerging some new collaborations. So thank you so much for joining, for being here. It was a pleasure to moderate this webinar, and I hope to see you next week. Thank you so much. Bye-bye.